I'd like to start with a quote that was recently sent to me by a colleague. Not a colleague in science, actually a colleague in professional services. And that quote was, "We owe our entire existence on planet Earth to a 6-inch layer of soil and the fact that it rains." And that really resonated with me, and it got me thinking about how we value soils. So despite their fundamental and intrinsic importance to human life, they are some of the most undervalued and underappreciated ecosystems on Earth. And we can see that in the way we've treated agricultural ecosystems. So estimates are diverse, but they all seem to coalesce to about a third of arable land being lost or severely compromised over the last 40 years due to erosional processes or pollution. And we know that soil is lost rapidly, but putting it back is a problem. Soil can be lost in a matter of years, but soil formation, pedogenesis itself, can, in some contexts, take millennia. So I think this represents one of the greatest threats to global agriculture, soil loss. And I think that's further compounded when we sit and think and calculate how much agricultural soil is sustaining life on Earth. 7.2 billion human beings are sustained by 7,800 cubic kilometres of soil. So if we were to visualise a cube of that dimension, that is 20 kilometres by 20 kilometres by 20 kilometres. That is all the soil we have to sustain the human population. And that 20 by 20 by 20 cube is going to have to sustain 10 billion people in the coming years. Now, I would argue that that realisation means that we have to place a lot more value on soils. We have to think about how we manage them in an agricultural landscape. Now, I'm a professor of soil biology, so I think it's probably no surprise to you that biology is going to be the focus of my talk, and trying to understand the interface between the physical world and the biological world. And I love this poster. I mean, it's a bit twee, isn't it? But it encapsulates the points from a series of EU projects that describes soil as the factory of life and explains why soil biodiversity is so important. Now, I think soil biodiversity is critical, and managing and harnessing soil biodiversity is central to the solution of soil restoration and soil protection in the agricultural landscape. Now, what I want to talk to you a little bit about is my pet, which is the mycorrhizal symbiosis. I've spent the last 20 years working on how plants interact with soil microbes, decoding the messages they send each other through chemistry in the soil, and understanding how they provide each other with resources and the processes that are involved, and how this leads to the protection and stabilisation of soils. So I want to talk to you about one particular symbiosis. This is called the mycorrhizal symbiosis, which simply means fungus root. It's a mutualism between a plant and its fungal partner. It's complex and varied in how it is initiated. Some incredibly complex signals are sent between the two organisms before they strike up a mutualistic partnership. And that partnership is underpinned by the reciprocal sharing of resources, of carbon flowing into the soil in return for the enhanced uptake of mineral nutrients. Mycorrhizas can enhance weathering processes in soils to liberate nutrients from rock. They can also do other things. So about 10%, possibly 20-30% in some systems, of all net primary productivity, so all of the photosynthesis that a plant is doing, ends up in the soil. And much of that occurs through the mycorrhizal symbiosis. So if these mycorrhizas are taking 10% of the carbon, that carbon isn't just staying in fungal biomass. That fungal biomass will turn over. We know that fungi exude complex carbohydrates that maintain and form the basis of the energy budget for what we call the rhizosphere, the community of organisms that live surrounding roots. So this energy can be used by bacteria. They can use that energy for fundamental processes, for motility, reproduction, and they also can use that energy to enhance nutrient uptake, mobilization of complex nutrients, mineralization, that kind of thing. So this complex system is being fed by plant carbon, flowing through fungi into the microbiome. We have characters like worms that feed and graze on the soil, particularly on these microbes, and they process that through their excrement. So we all know that soil is enhanced by worm poo, right? And it's these biological processes that feed into exactly what Steve was talking about, how soils form. Now if we have a look at the aggregation process, the process through which these macroaggregates that define soil functionality are formed, they're biogenic in origin. So we can see that roots and fungal hyphae are responsible for holding together some of the largest aggregates that we see within the soil. Those are then composed again of smaller aggregates which are biogenic in origin. They're held together by things like glycoproteins, so sticky proteins that are exuded by bacteria and fungi, and the kind of proteins that you find in the guts of annelid worms. And these themselves are composed of non-biological material, particularly clays, that kind of thing, but also smaller biological processes like humic material. And we know that these aggregates have different properties in terms of how they resist perturbation. So we know that the larger aggregates are very stable, they're not easily washed away, they trap moisture, they trap nutrients, and are responsible for underpinning the services that soils provide. As I said, these larger aggregates are biogenic in origin. So biology is fundamental and central to pedigenesis. And what have we done with soil? We've gone into the environment, we've created an agricultural system which is reliant on exogenous inputs. Those inputs are inorganic fertilizers, those inputs require extensive tillage, pesticides, and critically, the reduced use of things like lead. So the opportunity for soil to actually rest and recover from the pounding that we give it. There are some very simple processes that occur as a result of our interventions into the agricultural landscape. I mean, one of the simplest pieces of chemistry is if you expose carbohydrates in soil to organic carbon, the thermodynamic imperative, as a chemist would say, is for that oxygen to react with those carbohydrates and to produce CO2. So it's very easy to lose carbon, essentially the thing that's sticking soil together from the system by tillage. So I got very interested in the roles soil plays in, particularly in the processes that we're talking about, these inputs that have been defined by the Green Revolution. The Green Revolution occurred probably from the 1930s onwards, where our ability to harness novel chemistry and novel engineering underpinned a linear year-on-year increase in our ability to produce food. But interestingly, in the mid-90s, we've modelled this around about 1997-ish, coincidentally the year I came to Sheffield as an undergraduate student. Those year-on-year increases seem to either slow down or stop. And the yield, in this case of cereals, has plateaued ever since. When I go to speak to the wheat breeders, I ask them, what is the primary driver? Are we running out of genetics? And they said, absolutely not. The problem in their mind is the soil. So what do agricultural processes do to soil and soil biodiversity? In a landmark paper, a good friend and collaborator of mine, Thorin Helgeson, published this paper in Nature where she took all of the DNA that she could extract from an agricultural field and compared it to an adjacent woodland. She sequenced that DNA and she looked, in this case, at the diversity of mycorrhizal fungi. And she showed in this environment, there was a 55% reduction in mycorrhizal diversity, and she estimated a 65% reduction in the density of these beneficial soil microbes in a tilled agricultural soil. Now, I think that's, well, we now know that's probably a gross underestimate of the amount of damage done by tillage to soil microbial diversity. And this is where we bred our crops. This figure is one of the kind of starkest illustrations of those processes, and this comes from a project that was funded by BBSLC in collaboration with the White Rose Sustainable Agricultural Consortium. Some of you are involved. Steve Bamler is one of the major contributors to this project, where we've had a look at the distribution of carbon in agricultural soils, and we've had a look at the ability of those soils to sustain plant growth. This figure is actually from a master's student's thesis that kicked off some of these ideas a number of years ago, that was working with Jonathan Leake and myself in Sheffield. And he took soils from underneath a woodland, a hedge, a grassy strip, and then 232 metres into a conventional field. And he grew wheat in it. He matched the nutrients, so there's not a nutrient effect here, this is simply a soil quality effect. And he measured how the wheat performed. And he also looked at some fundamental soil properties. What he showed is that by the time you get 32 metres into an agricultural field, you've lost 70% of the productivity of the wheat crop, compared to growing that crop in a woodland soil. And this has absolutely nothing to do with the nutritional value of those soils. He also showed that by the time you move 32 metres into an arable field and you drought the crop, then the soil sets like concrete. So why? We see some of these fundamental characteristics of soils that underpin the services that they provide are being lost. We get a loss of soil organic carbon, we get a loss of large water-stable macroaggregates. And if we have a look at how these factors relate to growth, we can see, for example, that water-stable macroaggregates are powerful predictors of plant performance. So how are we going to fix it? Well, if we want to get carbon into the soil, we need to vector it in there in some way. Mycorrhizas are a good way of doing it. Unfortunately, having bred our crops in soils denuded of mycorrhizal inoculum and having provided those crops with everything that they need, so there is no advantage to using those mycorrhizas, we have accidentally selected against the mycorrhizal trait, for example, in wheat. So these are cultivars up here that are a little bit older in the pedigree, and pretty much everything we commercially grow sits in that state. So modern varieties don't make mycorrhiza. We developed this project to try and use mycorrhizas, establish the mycorrhizal trait within crops, and use that as a way of restoring soil. The way we've done that is we need to actually restore the mycorrhizal populations that are present in the soil and reconnect the genetics of the crop to those populations. So here's our field trials. This is a simple ley. We're using a ley to help recover soil structure and soil microbiology, and we're comparing that to conventional wheat production. This is all up at the Leeds farm. Let's have a look at some indicators of recovery. These are some early data that we're getting, looking at things like earthworms, which we know are critical to soil recovery. And after 2 years, only 2 years in ley, we're seeing dramatic enhancement of populations of worms. These data came from Joe Holden's group up at Leeds. And we're seeing an increase in water-stable macroaggregates if we're using a ley strip, even compared to minimum tillage treatments. We're seeing a reduction in the smaller aggregates as those aggregates are converted into larger aggregates through the biological processes under the lab. In terms of the ability of these systems to resist environmental stress, let's have a look at drought and water holding capacity. This is the water holding capacity of soils taken from ploughed, min-till and 2-year ley strips in our field trials. We're getting about a 10% increase after just 2 years in the water holding capacity of our soils. And in drought trials we've shown this gives you several days before drought symptoms occur in your crop. So what do we need? We need a new model for agriculture. We need to start thinking about how our agricultural system operates for us and how we use it in space and time. I don't have a silver bullet, and I don't believe there is one. But I think we need to make some brave decisions about how we manage the agricultural landscape and start to think about potential extensivization, certainly spatially, of the agricultural system, the amount of land that we might be able to use in agriculture, and think about intensification temporally in terms of the land that we actually do use to support food production, to allow us to increase the amount of time that our soils could spend in recovery. So one of those ways is to try and get organic carbon in there. It's one of my lessons of public engagement. If you open your mouth at the wrong place at the wrong time, you end up at the front page of the Metro with a headline like that. But I stand by it. We need to think about using alternative fertilisers and integrating organic carbon in any way we can into the agricultural landscape. I'll leave you there with a quote from good old Franklin D. Roosevelt. Lest we never forget the nation that destroys the soil, destroys itself. Thank you.
Thanks, Duncan, that was great. Could you speak a little bit about the final slide? You were talking about the central aspect, and on the one hand you have this wonderful effect from the slowing down of agriculture, from the delays, but on the other hand you're talking about an intensification. So could you unpack that a bit?
I think what we need to think about is how are we going to get enough food to feed 10 billion people? Now, we could plough up all of the land that we've got, trash the soils, and effectively mine the soils for their resources, and that will work for a while. But we know that if we leave our soils alone, they will recover, they can get better, which means we need to have some sort of system that is temporally flexible enough to allow us to maintain agricultural food production, but allow our soils to recover. So if we, as the kind of sustainable intensification model, which I think is a bit of a contradiction in terms, the sustainable intensification model tells us we can have our cake and eat it. We can intensify the land that we have, and we can produce more food. And I think that's up to track. I think if we're going to continue with an intensive agricultural system that we probably need to produce the amount of food that we're going to need for 10 billion people, then we need to have a think about where and when the land that we're using to produce that food occurs. So maybe we need to think about an agricultural landscape that is much bigger, that we look at the artificial landscape that we have created, certainly in Northern Europe, the pastures that are effectively now non-agricultural, reintegrate that wider agricultural landscape into food production. And we need to start thinking about coupling that in terms of mixed use, coupling pastoral agriculture, pastoral processes and agriculture together. So we've got an integrated system that really takes advantage of the kind of opportunities of food production, but also recognises that our agricultural landscape has pretty much been maxed out. And the only way we're going to see significant recovery and a reduction in the kind of inputs that we're going to need is through allowing it to rest and recover. And that simply needs time.
But you keep saying we need to. Who are the we, and how do you operationalise this change in agriculture?
The we, I think, is all stakeholders, from scientists to farmers to policymakers. And I think one of the things that I've enjoyed so much about heading up a interdisciplinary translational research institute has been the work I've done spanning social science into arts and humanities, working with stakeholders and using clear science. So one of the things that I believe very strongly is that we probably got the technology we need to solve the problem. We need the political and social will to enact it. And that's why pure and social science needs to work together in this kind of way in order to enact those solutions. So I think stakeholder engagement and co-design is incredibly important. So I'll give you an example of a piece of work that I've done in Brazil, where we were working with a series of farmers on optimising wheat for a particular landscape called Cerrados near Amazonia within Brazil. We had some ideas about how we might improve the wheat varieties that they could use and how we could create drought tolerance in a usable way. If we'd have gone in with a project and tried to do that without having spoken to the local community, we'd have created a wheat that would survive that no one would have grown, because the cultural properties, or the properties of the grain that were embedded in the local culture were such that the kind of backgrounds we were going to use would be totally socially unacceptable to the farmers, and actually would be not compatible with the agricultural processes in that particular region for red-fed agriculture. So instead, we spent a long time co-designing the scientific approach. Actually, the science experiments were designed 50%, I think, by input from the local community about how we might align modern genetics with traditional growing practices. So I think that really highlights how important it is that, you know, this is not, social science does not come at the end of these projects. It is embedded at the start, and you use co-design principles in order to define pure scientific research.
The two questions here, Anne and then Amartya.
I'm just wondering whether you could talk to the issue of mycorrhiza and food quality, because that's a possible motivator for a transformation.
I didn't have time to come on to that, but we've done quite a lot of work in my lab on the role of soil microbiology and biofortification. So particularly if we think about the kind of things that mycorrhizas do, weathering processes, it's not just macronutrients that are cycled through healthy microbial populations, it's micronutrients. I had a master's student recently in my lab show that particularly selenium deficiencies can be overcome by highly mycorrhizally competent cereal varieties. So we're doing some work through some GCRF priming funding that we have in East Africa, where we're looking at the role of soil microbiology as a solution to hidden hunger. So that's very much again a social science, pure science collaborative project.
Amartya, and then Bruno has a question.
So in the context of urban agriculture where food is grown indoors and on the rooftops, one of the problems is rapid nutrient depletion. Do you think that, to what extent do you think such systems can be incorporated, if at all they are useful?
They can be incorporated, but it's not easy. So it's almost like people have placed questions for me, because that's quite a nice question, because I've just had a new project funded, which is developing synthetic soils for urban food production and incorporating microbiology within synthetic soils that allow the food insecure in urban areas of North Africa to grow their own food locally.
Well, that actually answers part of my question, which is your 2 years sort of resilience of the soil is associated with soil which are not completely desegregated, because we are also told it takes centuries to get soil.
Absolutely.
But would you bet on artificial soil?
I think artificial soil is a partial solution, absolutely. The critical thing is that it needs to be recyclable. So I think current substrates are environmentally unsustainable. You look at things like coir, you look at things like rockwool, you know, they end up in landfill, and that is completely unacceptable. So one of the things that we've been doing is using biological oils as substrates to produce polymer foams, oils, non-mineral oils. Mm-hmm. So, and also petrochemicals. We've done life cycle assessments to look at the carbon embedded within the production of infinitely recyclable polymer foams as synthetic soil. So if you can design a soil that is based on plastic, a plastic which can be recycled with very little energy input, then you can produce a very sustainable system for local food production. So to say I had an EPSRC proof of concept grant to develop that technology with my colleagues in chemistry, and we're now deploying that in the MENA and in the Gulf region to have a look at food insecure communities in North Africa and how we might use that and how they might locally recycle those polymers.
We might avoid the perfect storm.
I think we can avoid the perfect storm, but I think we have to use...
The only reassuring thing, you know.
We have to use technology, but we have to use it responsibly, and we have to really think about how kind of the social context of deploying that technology.
Danny?
Can you talk a bit about your human project? Is that part of building soil? Is that part of artificially building soils?
Yeah, so it's called the SHIP project. Literally, that's what we called it when we got it funded. And that is looking at anaerobic digestion and the role of producing organic fertilizers, but processed organic fertilizers. One of the things we're interested in are particularly complex phosphates, which are plant unavailable, and how we can use microbial processes within formulations of organic fertilizers to enhance phosphorus cycling. The nitrogen we can deal with, but phosphorus is a problem. And we're developing some technologies in Sheffield jointly between my lab and the chemical engineering department to address some of those problems.
Christina?
Yeah, I have a question about, I mean, the scale of the 10 billion, feeding 10 billion. I mean, I don't, because I see that when you scale down in every case, it's a case. You're in Northern Africa, you're in Brazil, you're in, and so actually the farmers that I work with, they say that's the wrong question. It's not a question about feeding 10 million people. I mean, that's not even the conception of what we should be asking. It's about how in each place, like what is decimated food.
Who's we?
I'm saying, what?
Who's we in that context?
How, did I say the word?
The farmer that you work with.
Sorry? Oh, I'm saying.
What I don't understand is, because I agree with you that there are so many different scales at which this operates.
Yeah, so I guess I'm just asking, like, wondering how you work with those scales, because it does seem that there's this looming question that's a very planetary scale, but then actually it really situates in specific contexts of the question of food autonomy,
local
sovereignty.
solutions to global problems is a really important concept. And I think we do have to look at, there are local, we can learn a lot from local microcosms about how we need to treat the global problem. But we need to very clearly think about the kind of interventions that we make when we think about the scale at which those interventions can be applied. So what I think the scientific interventions can be pan-globally important, but they may not work in particular cases. So that's where I see the interface with social science and pure science is so important. I can create a technology that could solve problems in several continents, but it might not be socially acceptable to deploy that technology. GM is an obvious example of that. So I think what it's up to us as scientists to do is to create the technologies that can solve the problem, do that through co-design, of course, but also understand the context in which those solutions will be acceptable or not, and come up with create a toolkit that we can then draw upon to solve local problems. That's how I see it working.
Well, I guess just there's assumption of a universal nature and a diverse social. And I think it's not just that it won't be socially appropriate, it's actually that ecologically it's not viable. Like there's not a singular nature that it can.
Absolutely, there's no magic bullet, which is why I think about it as a toolkit, an arsenal of bullets that can be deployed in combination. But how those are deployed in which combination will be a question for I think, local issues. And there will be as environmental, there will be also as specific as the kind of soil that you're working with. But that toolkit we need to think about as technology is developing, and then working out how we can deploy it best. And if what we're missing from that tool.
I'm just going to have a last question from Steve, and then let's go and have some lunch.
Yeah, I think it's just in the spirit of debate here. I think that the perfect storm question, is about, it sets this scenario that potentially, we're looking at somebody taking a really big hit. Mm-hmm. And the question is who and where. And then a colleague who works in international development says, well, the perfect storm is a great Eurocentric concept, it's all in the future, but there are many parts of the planet where the storm now rages and others where it has already passed through. Yeah. And I think the perfect storm, that's really important, is to think of that. And so it is, that comes a little bit to this point about where is it going to hit? And it acknowledges, we have to acknowledge that it's raging in some places.
Thank you. That was a great note to end the presentation part on. At 1:30, so I'm pushing the program a little bit ahead so that we have more time